Hi, I'm Josh. This is my physiology final on ribbon worms. If I stumble or stutter and I don't edit it out, that's because I have to do this all in one take, just the way that the my phone sends it. It doesn't let me do segments. So if that happens, that's why. But anyway, ribbon worms. A ribbon worm is any worm in the Nermitae phylum, and the reason why they're all grouped together in this phylum is because they have a specialized appendage called the proboscis, which is what these slides are about, the proboscis. But we'll get into that later. These worms, they vary in size greatly. The smallest of them are just a few millimeters, and the largest of them can grow over 15 meters. And they mostly live in shallow ocean water, but there are a lot in the deep ocean, rivers, lakes, and there's like less than 20 species that have adapted for land as well. But we're not going to mainly be talking about them because I'm sure they're different than the ones in the water. What is a proboscis? If you look right here, that white thing coming out of a worm attached to the hand, that's the proboscis. It kind of branches out. That's because it's part of a specialized class within the phylum, which we'll go into later too. But a proboscis is a hunting appendage or a defense appendage. It rests within the worm's bodies, and it gets shot out of their bodies when needed. It's composed of muscle, and it's wrapped in an epithelium. The epithelium has gland cells, and those gland cells contain mucus. And once the proboscis exits the worm, the vesicles bind to the gland cell wall, and they get released onto the proboscis, covering it in sticky adhesive mucus. Where is the proboscis? So the proboscis itself is within a sac. It's a fluid-filled sac called the rhinoceral sac. If you look right here, that little U, that's the proboscis. That white blank space, that's the fluid within the sac. And then right along the edges right there, you can kind of see like a darker ring, like a darker red, pink ring. That's the epithelium of the rhinoceral sac itself, and surrounding muscles that help it contract. So the rhinoceral is either connected to the mouth itself or its own specialized orifice right above the mouth, so that way the proboscis can exit the worm. And as you can see, the proboscis is just kind of like free floating here. It's not attached to anything, but that's because in the back of the worm, like right over here near the back, it's attached to a retractor muscle that helps it bring it back in. The fluid itself is an extracellular fluid that's mucin rich. All right, how does the proboscis work? If you look right here, you can see the proboscis exiting the worm after it gets booped with a stick. It branches out, which is pretty cool. There are 4 stages to how this happens. Stage 1 is sensory, sensing it, like a reason for the proboscis to come out of the worm. Here, that's the worm being hit by a stick. Stage 2, eversion, which is what this video shows very well, is it being averted out of the worm. And stage 3, grappling. When it grapples its prey, it gets entangled in the branches or gets like wrapped in the proboscis. And then stage 4, eversion, when the prey gets captured, when it gets brought back to the worm. All right, stage 1, sensing. There's specialized cerebral organs, and what those are, they're components in the head. There's one on either side of the head, like, like, think of it like right there and there, pretty much. And what they are is these organs contain chemosensory cells, and those chemosensory cells are exposed to the outside water. And when what those chemosensory cells do is they detect small molecules within the water, like dissolved molecules, like a prey's mucus trail. And it will follow that trail, and once the concentration gets high enough, the cerebral organ will send a message to the worm's brain, and then the brain will send a message down the ventral nerve cord, which is the worm's equivalent of a spinal cord, to the rhinoceral sac. And once the rhinoceral sac gets that message, it contracts. which brings us to stage 2, eversion. So the rhinoceral sac and the muscles around it, they receive the signal, they contract, and when that happens, the pressure in the fluid, it rises. Something in the system has to give, and that's the proboscis exiting the worm. The proboscis itself, it rests within the worm inside out. That's why it has to be everted. And then once outside the worm, the proboscis can expand up to multiple times the worm's body. It's very, very stretchy. Stage 3, grappling. So the phylum itself is separated into 2 classes, and those classes are different because of the way the proboscis works. So on the left here, the Anapola with an A, the proboscis branches out like the ones that we saw previously, like spaghetti. And the mucus on both of these actually is adhesive, and in some species, it's toxic. It carries a toxin. We'll get into that in the next slide too. But the Anapola, it wraps around the species, it entangles it in the spaghetti, and then it retracts, but that's the next slide. And then in Anapola species, the proboscis does not branch out. It's one straight noodle. And it has spikes on it sometimes called stylets. They're just calcium-rich, hard spikes. And there's sometimes one, there's sometimes multiple, depending on the species. But what that does is the ribbon worm, it will stab its prey and get a good grip on it. And when it stabs it, the toxin gets into its bloodstream faster. And stage 4 is retraction, which is that retractor muscle we talked about. It contracts, and when that contracts, it pulls the proboscis back in. Right over here, you can see like one long pink noodle. That's Anapola. That's like what the other species. I couldn't find one that had spikes on it, but just imagine that with a couple spikes. All right, and then this toxin is where things get kind of cool. There's 3 ways the species can have a toxin, which is kind of interesting, because the first kind is they transcribe and translate it by themselves. It's a peptide toxin. It's about like 50 amino acid lengths long. And the way that this works, and all 3 of these works, is that it causes paralysis by binding to the sodium channels on nerves. So the sodium channels can't function, action potential can't be reached, the organism can't move. All right, the second kind of toxin is produced by a symbiotic relationship between bacteria and the worm itself. That's called a tetrodotoxin. And then there's anabasin. It's produced by the worm's own metabolic pathway, so it can take something else and then produce anabasin from it in a series of steps. But all 3 of those work by acting on the sodium channels, inhibiting them, and causing paralysis. Uses. All right, so the peptide toxin I just talked about, it's highly effective in small doses in insects, but it has very low toxicity to human cells. Because of this, it's being researched as a biopesticide, excuse me. And I couldn't find any examples of this. However, if there's any engineering majors who are interested, I think it would be pretty cool to see the proboscis model being used in soft robotics. Making something based off the ribbon worm. And then unknowns. We don't know how the anabasin pathway works. There just hasn't been much research on it at all. There hasn't been a need to research it. So there's not much information out there. But the neural circuitry as well has not been fully decoded. So we don't know how the cerebral organ understands the chemosensory cells. We don't know the communication going on there. The same way that we don't know the communication going on between the brain and the cerebral organ. We don't have the neural circuitry worked out. But that wraps up my final presentation for physiology. I hope you liked it, and have a great day.